Hello students, welcome back to a new video of Sunanda's tutorial. So in this video we will discuss about X-ray crystallography. What is X-ray crystallography? So X-ray crystallography is an experimental technique or analytical technique that uses the X-rays. Here X-rays are diffracted by crystals, particularly crystals. It is not an imaging technique. This technique here, in this technique, X-rays are used. X-rays have the proper wavelength. That proper wavelength are to be scattered by electron cloud of an atom of comparable size. And based on these diffraction pattern obtained from X-ray scattering of the periodic assembly of molecules or atoms in the crystals, the electron density can be reconstructed. Okay. So here only crystals are used. Crystals in X-ray crystallography. X-ray crystallography is a technique that uses X-rays to determine the atomic as well as molecular structure of a crystal by analyzing the diffraction pattern. So, when we use X-ray, the rays are scattered. So, it analyzes the diffraction pattern created when X-ray interact with the crystal's atom, interact with the atoms present in a crystal. The principle, at first diffraction. So you have to remember these points. At first diffraction, then 3D structure, three-dimensional structure, then electron density map. So at first diffraction, X-rays with wavelengths comparable to atomic spacing are diffracted or scattered by the atoms in a crystal, creating a pattern of spot. How many atoms are there? How they are placed inside the crystal? Then 3D structure. The angles and intensity of these diffracted beams contain information about the arrangement of atoms within the crystal. Then electron density map. By analyzing the diffraction pattern, a crystallographer can create a 3D map of electron density within that crystal, which reveals the atomic position, the position of atoms present within a crystal. Okay. Next, what are the steps of X-ray crystallography? At first, crystallization. The molecule or protein of interest, the sample, at first we have to crystallize that sample. It must be packed in a regular, repeating three-dimensional lattice. Then X-ray diffraction. A crystal is exposed to a focused X-ray beam and resulting diffraction pattern is recorded. Okay. So at first a crystal is exposed to a focused X-ray beam. So a beam of X-ray to be used or focused on the sample, on the crystal. Then data collection. The diffraction patterns are collected and processed, often using specialized software. Then structure determination. The diffraction data is used to solve phase problem, calculate electron density, which reveals the atomic position of atom inside that crystal. Then model building and refinement. So a 3D model of molecule is built based on the electron density map and refined through computational technique. So here only crystals are used because crystals are necessary because it neither possible to see or handle a single molecule by this technique. So crystals are used because inside the crystals a lot of molecules are present. Okay, a defining feature of crystal is that it is three-dimensional periodic array of molecules. Here all the molecules in the crystal occur only one or in a few orientation. This leads to a tremendous amplification of diffraction image, and not only that, the diffraction image is also much simplified. Okay. So why we do crystallography? The knowledge of accurate molecular structure is prerequisite for some drug designing. The knowledge of accurate molecular structure is required, sometimes is necessary to discover a new drug. Okay, next. So this is the structural representation of different steps of X-ray crystallography. The diffracted X-rays are scattered by the crystals at certain angle. So after the bombardment, the X-ray on the crystal, it produces the diffracted beam at a certain angle by producing certain angle. The further backward the X-ray scatter, the higher we say the resolution of the data set. The extent to which crystal diffracts determines how fine a detail we can actually distinguish in our final model of the structure. So high resolution is very necessary here. Knowing the wavelength and the diffraction angle of a reflection, its resolution D can be easily calculated by Bragg's law, which is n lambda equal to 2D sine theta. Okay. So high D is necessary. Okay. To produce the final model accurately of the sample, and D can be calculated from this equation, which was first provided by Bragg's equation. Okay. Next, application in structural biology field, determining the three-dimensional structure of protein, nucleic acids, DNA or RNA, and other biological macromolecules. Then material science, in the field of material science, this technique is used for characterizing the atomic structure of materials including metal, mineral and semiconductor. Then in the field of drug discovery, in the field of medicine, understanding how molecules interact with biological targets, which is crucial for drug design. Then materials characterization, for identifying and understanding the structure of new material. Okay, next. So it is all about X-ray crystallography. I hope you will get benefit from this video. So if you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.